So I'm going to tell you about work that we've been doing in molecular bases of adaptation. And it starts with some of the stuff that Andrea was involved with, like Klaus mentioned, and that kind of sets the stage for things that we've been doing since then. And I'll focus on the, Cape Verde populations, but kind of mention some of the other interesting ones. Okay, let me see if I can move this so it's not covering. There we go. Okay. So we're interested in understanding adaptation in natural populations, and there can be a lot of variation even within the same species in color and size, things that are really obvious to the eye, but also less obvious traits related to like biochemical traits or molecular traits. And understanding how populations adapt to local environments can help us understand the past and predict what kinds of changes in time frames would be necessary for adaptation to future challenges. Not quite figured out how. Okay. Organisms confront at the same time multiple types of environmental challenges. So sometimes there could be kind of interplay between the ways that organisms adapt across these different types of environmental challenges that could be either kind of competing in networks to adapt, or there could be pleiotropy, where you have adaptations that are occurring from some variants to multiple traits. Model organisms and mutants in model organisms have played a key role in biological discovery. So people used model organisms and conduct mutant screens, and that's how we know a lot about the structure of pathways in model organisms. But there's also limitations to what we can learn from mutants in model organisms and mutational screens. Natural variation can be more complex, and you might have multiple mutations that occur, kind of co-occur across space. You could think of natural variation as kind of like a nature's mutational screen, where you have mutations arising across space, and mutations accumulate over space and time, and the different combinations can be giving you different traits and trait combinations. But then with those, there's also the challenge that you need to work backwards from this kind of variation in nature. It can provide these combinations and also the ecological context. So when you do mutational screen, you don't really know what those trait differences, if they would have any ecological relevance. But once you kind of cross it through with with what you can learn from ecology and evolution, then you can understand the functions of those of mutations and genes. So thaliana is a powerful model. It's the predominant molecular model plant. So we know a lot about the genome. We have a lot of tools in Arabidopsis thaliana. It's naturally inbreeding. So inbred lines represent what you see in nature. You don't lose much because they're inbreeding somewhere between like 85 to 99% in nature. So they've kind of it's not like in a mouse or something where when you inbreed them you lose a lot of the variation, or in corn or maize. And then you can use those inbred lines for lots of different things, and then have that information that you can line up across those. So you can use it for phenotyping in lots of different environments, and sequencing and follow-up functional analyses. Yeah, and there's a lot of information and gene annotations from the work people have done, often mutant screens. A lot of thalianic accessions have been collected in the last 100 years or so, and most of these collections have come from the Eurasian range, which was thought to be like the whole of the native range, as shown in this map from 2012. And this has been a vital resource for mapping. So using the natural populations, either in recombinant inbred lines or in genome-wide association studies to map the genetic basis of our traits. But historically, the sampling had a strong Eurocentric focus. So you can see most of the yellow. I don't know if you can see if I put a laser pointer. So most of the collections are in Eurasia, and especially in Europe. And this is because this is where most research was focused. Most of the researchers were living there, and also this was kind of thought to be the native range of the species, because for the genus, this is where the genus tends to be concentrated in Europe and Central Europe. These populations outside of Eurasia, in particular the African populations, hadn't been studied much, and they were thought to be recently introduced. We got interested in these actually for a Cape Verde project that I'm going to tell you about more later. But we wanted to find the ancestor there. We started sequencing what we could find in Africa. So there's one accession from Cape Verde, and then around 70 accessions from Morocco, and some herbarium accessions from Algeria, mostly herbarium accessions from East Africa and from South Africa. And this was done by Andrea Fulgioni and Arun de Vasula. And what we found was really surprising. So we expected that if these were recently introduced, they should be kind of a subset of the Eurasian variation. But instead, we found that there were deep reticulated branches in Africa relative to Eurasia. And Eurasia is almost looking like a star-shaped genealogy where there's not a lot of reticulation. It's kind of like a more recent rapid expansion. So this was really exciting and surprising finding. When we look at the principal component analysis, we also see that the first principal component separates out African from non-African populations, and also the second principal component separates out those North Africans. And the 1001 genomes is mostly represented here with a little bit of this variation from Eurasian relics. So there's a lot of new variation that we found in this really well-studied species, and that gave us kind of something further to look at. This is published like 10 years ago, so I'm not going to go into the details, but we did some coalescent reconstruction and also JSFS to see if it agreed, and looked at the timing of splits, and we got this kind of model. We have very little information from South Africa and Tanzania, right? We had these herbarium accessions. But we saw it looked like the deepest splits within Africa, and then Europe and Asia split off from there. I'm not going to go a lot into our additional sequencing, but this is kind of, there are some old lineages in Asia that are leading down into this sub-Saharan Africa clade we know now. Okay. So while Arabidopsis is growing broadly throughout Europe and often in disturbed habitats along railways and near buildings, it also grows in natural environments in Morocco and Cape Verde Islands, Canary Islands, East Africa and South Africa. And so there's a lot of variation that hasn't really been explored, and a lot of kind of regions where there could be pretty harsh environments and potential for adaptive evolution. So we've gotten together with a lot of collaborators from different regions to try to sample some of these undersampled regions and use those to study the history of the species and also trait variation and adaptation. So this is showing the extended variation map that we've sampled more in East Africa, especially a little bit in South Africa, Canary Islands, and a lot in Cape Verde, which is published. And then we've resequenced some of the accessions that have been skipped before. So we've resequenced some more accessions from Asia and also from some of these far northern populations outside of Sweden. Sweden's been studied a good bit, but we've got some from Norway. We had some from Finland, but I think they weren't Arabidopsis, so you just said Norway at the end. They weren't thaliana. Okay, so yeah, so this is our kind of revised tree. We have this East African population is very distinct from the other guys. The outgroup is some kind of probably an old population from East Asia, and and Asia is kind of a population that comes out of Europe, but is also has distinct lineages. This is an old split. And then we have these other Moroccans are kind of spread around, and Cape Verde. And let's see, this has been, Sophia Sacco Ziri has been working on this population genetics in here. We have a tree that we rooted. This is just neighbor joining tree, but we rooted it by an outgroup that's the other Arabidopsis species. So sequences from Halleri and Lorata and Arenosa. And from that, we have Morocco. The Moroccan Riff is coming out as the kind of closest to the root, followed by Madeira and the Iberian relics. And what we had found previously, not to get into a lot of details on it, but there's an S locus in Arabidopsis that is kind of a relic of the transition to selfing. And before they transitioned, they had lots of different S locus haplotypes. And then when selfing started, they would have lost those. And most of the populations have really limited diversity. In Europe, there's mostly haplotype A or AC recombinants, and in other populations, some of the more, like the East Africans are all haplotype C, but we have all three S locus haplogroups in the Moroccan Riff. So we still think that this kind of best represents the ancestral population of Arabidopsis. So, and for sure, these kind of deep branches in the root seem to represent the oldest variation in the species. And so now we can use these to try to understand the genetic basis of different kinds of traits in these other populations and worldwide. And so this makes a new resource for understanding adaptation and looking at kind of molecules to ecosystems where we can look at different traits and different levels of variation. And we collaborated with some groups to do phenomics on these. So Mark Arts on photosynthesis phenomics, Olivier Ludet, drought, and David Salt for elemental content. And then we're trying to kind of reconstruct these different genetic architectures of traits. And I'm focusing, well, today I'm going to focus on Cape Verde, but we were maybe quicker to publish some of the adaptation related traits, the traits where there's really strong signals of adaptation and kind of simple genetic architecture. But one thing that I'm interested in is getting into more of the kind of variation in genetic architecture across traits also. Okay, so in Cape Verde, we're looking at adaptation after a kind of rapid shift in selection where they've colonized from the mainland to Cape Verde. And this is in collaboration with Angela Marina, Anita, and Herculano Dinish at well, he was at the national parks in Cape Verde when we started the project. So this, we thought, because this might be a simplified population relative to the mainland, that it could help us to identify precise functional variants that underlie adaptation in nature. It might be easier to get to those. These continental populations are complex with different kind of refugia and mixing after the last glacial maximum, which makes it difficult to identify causal genetic variants, and then also difficult to reconstruct their histories. And islands can represent natural laboratories where complexity is reduced. The theory of evolution originates in islands, so they're relevant for understanding evolution. And island species are also interesting because they're vulnerable. So understanding how island species have adapted and how that adaptation, or how they've evolved really, and how their evolutionary histories might impact their future risk is interesting. So there are a lot of endemic species with narrow distributions, and these are at increased risk of extinction relative to continental species. And since the Industrial Revolution, island species lost genetic diversity at a rate almost 5 times greater than the continental species. So they're at risk. Okay, so CDI0 was an accession that was interesting to the Arabidopsis community, and And one thing that people like to do in Arabidopsis is to take representatives from across the thaliana species that have been collected, and then just do phenotyping and see how the phenotypes compared. And CVI0 was often an outlier. It wasn't clear how it ended up there. There was just this one accession that had been collected and was available in the stock center. And well, it seems to actually have come through Cologne, but and through George Copeland and Martin Corneef, but nobody's quite sure which botanist donated it. And it was used a lot, so we know a lot about it. And it's also, because it was kind of geographically and climatically distinct, it was a question of where it came from. So what's the history of the Cape Verde Islands? They were colonized by the Portuguese around 500 years ago, so around 500 Arabidopsis generations. The current flora is a mix of endemics and species that were introduced since colonization. So people thought probably Arabidopsis in Cape Verde was introduced more recently, and that it's just moving around easily and not endemic. And the main inputs of endemic flora are coming from Africa and North Africa and the Canary Islands. It was also the first stop on Darwin's Beagle tour, and he didn't stay very long because it was a dry, kind of desolate environment, at least the part that he visited. He said in his diary, "My imagination never pictured so utterly barren a place as this. It rains, but very seldom in this country. And when it does, a mass of vegetation springs up. This soon dries up and withers." So this points to what's probably one of the main selection pressures in Cape Verde, which is that it's got this long, extensive dry season. So for the plants, they need to grow and flower quickly when the resources are available, and try to kind of finish their life cycle before the dry season starts. And so, like I said, it's been studied. CVI0 has been studied a good bit from different labs, and it's been found to have divergent phenotypes. It's fast flowering. It's got a reduced circadian period, strong seed dormancy, low water use efficiency, low gene body methylation. It's flooding intolerant. It's something called a shade avoidance syndrome. So it kind of seeks out the light, divergent mineral accumulation, and divergent light response. So this divergence in traits suggests that there's been adaptation to a divergent selective regime. And so we thought that this kind of combination of an island system and a model, which CVI0 is kind of a model itself, would be a useful system for reconstructing adaptation. So when I went to collect, this is when I was still in Vienna, I went to collect Arabidopsis, and the second trip also Andrea went with me. And what we found, the first trip was with Fabrice Roux, actually. What we found was that Arabidopsis was not in kind of disturbed habitats, but it was with together with native vegetation on these rocky outcrops, which represents the ancestral habitat for the genus, for the Arabidopsis genus. It tends to be found on these kind of scree slopes and rocky outcrops. And it's present in 2 islands in Cape Verde, and these are the 2 islands with the highest altitude. And it's only found at altitudes of around 1,000 meters and greater. And these are Santa Antão, and Fogo. And yeah, so it's just those 2 islands. And the habitat suggested that plants had been subjected to potentially to a multivariate environmental shift, and that combined with what we know about CVI0. In the habitat, we have long dry season with rare unpredictable rains. Moisture that's mainly coming from trade winds and fog, volcanic soil, high ultraviolet light, and they're often germinating in shade, with reduced seasonal variation in day length. They're closer to the equator, so the day length is similar year round instead of so such a large difference in the summer and the winter. And they have a high growth angle on rocky substrate. So a lot of these things fit with some of the phenotypic divergence. So we work to try to reconstruct population history and then infer trait architecture and the adaptive history. We sequenced a few hundred accessions from Cape Verde, and then we could compare those to accessions from Madeira, North Africa, and the 1001 in Eurasia. And we found that CVI looked like a single colonization event. So it's derived from of the sampled populations we have, present day populations, it's closest to Morocco, but there's still a long branch that separates it from any individual from Morocco. I think I'm not going to get into a lot of the modeling we did there, but different chunks of the genome are closest to CVI across Morocco. And so in the end, we inferred there's some kind of a kind of ghost population that's no longer around. It could also be Canary Islands, but Canary Islands has had lots of introductions. So similarly, we don't have like an individual that's closest to CVI, but it looks like they're coming from the Moroccan clade. But within CVI, we see kind of a distinction between the two islands. And relative to this Moroccan outgroup, the diversity in Cape Verde is low. It's a couple orders of magnitude lower in each island compared to Morocco. So there's about 8 variants per 100,000 base pairs in San Antao and or 100 kb in San Antao, and similar in Fogo, 9 variants per 100 kb. When we look at the joint site frequency spectrum between Cape Verde and Morocco, we see a lot of variants that are, these would be, okay, so the joint site frequency spectrum, if you collapse along the vertical axis, it gives you the counts of variants of singletons, variants that are found once, twice, 3 times, or fixed in the population. And then if you collapse along the horizontal axis, then you see the counts of variants that are segregating in Cape Verde, and then these variants in this internal part of the plot show you shared variation, and what we saw is basically no shared variation. So from like the phylogenetic kind of level, these are different species. These look like different species. And similarly between islands, let's see. So yeah, so you see the counts of variation in San Antao and in Fogo, and not shared variation between the islands. So it implies that there are strong colonization bottlenecks in the origin of the islands, and these kind of produce a clean slate for adaptation, where adaptation would need to occur from any adaptation that occurs after colonization would mainly come from new variants that arose after colonization. And these populations are analogous to endemic species, where you have strong bottlenecks between populations, and you know, depending on which speciation concept you want to use, like if you can cross the populations, there are some incompatibilities that we're interested in, but this is maybe also within islands. And then also I'll talk about one of the variants that would cause an incompatibility between Cape Verde and the other or Fogo, the Fogo Island, and the other worldwide accession. Due to selection, I guess. We haven't presented it that way, but okay, so we inferred the history of the populations using a coalescent-based approach. We used, what I'll show you is with Relate, I believe. And so it's using information from across the genome from these haplotypes that are separated by ancient recombination events, and using the variation in each region to get an estimate of the effective population size, which is expected to be inversely correlated with the time to coalescence. And so when we look at this, we see, and we looked at the ages of the splits of the islands in lots of different ways. It gets messy when you look at the split of either island to Morocco, but when you look at the split of the 2 islands to each other, there's not a lot of incomplete lineage. So there's not really any incomplete lineage sorting. I showed you the JSFS. So, so no matter how we do it, we can do a back of the envelope calculation and we get something pretty similar in terms of split times, but it was around 5,000, say 3,000 to 5,000 generations ago. And then this is showing that the populations, this was what we infer to be colonization, and then populations expanded. So this is showing Saint-Antal and Fogo, and if you kind of plot, make pseudo-diploids across the two populations, then you get a faster expansion. That's due to their divergence from each other. All right, and so CVI islands were colonized through severe colonization bottlenecks. There's a bit more ancient structure in Saint-Antal. Fogo has this long period where nothing really happened. It seems like it was either a very small population or it grew and most everything went extinct. And then it's about 900 generations where it looks like a population of like 40 individuals, basically. There are a lot of fixed variants in Fogo. The signature of that is that there are a lot of fixed variants in Fogo that appear to have happened at that early time. And, okay. Okay, so then we started asking, before we mapped, we did a few things. I'm just going to talk about one here. I kind of added this in because it went a little bit with what Pauline was talking about. So we looked for evidence that there was differential fitness between CVI and Moroccan accessions, with the idea that, you know, this is a bottleneck population, and so is there some kind of evidence that the population just in general doesn't do as well? It has lower fitness because of some genetic load or something. I think that's what I'm going to show you. Yeah, we simulated the CVI conditions and we simulated Moroccan conditions in a growth chamber and based on data loggers that we had placed in the field. And when we grew them, now the Moroccan conditions we used were a pretty easy condition. For sure, we could use a condition that's got really more cold to if there was a more of a kind of freezing. They might have more freezing in some of the Moroccan populations, so it's kind of an easy Moroccan, but we didn't see a big difference in the Cape Verde fitness compared to Moroccan in this kind of basic Moroccan condition, showing that the Cape Verdes don't look like they've really fallen to a low fitness in a simple condition, but they perform much better in this CVI simulated environment. And we think anyway that Arabidopsis probably, I mean, there's good evidence from when you have really strong bottlenecks for island foxes and island wolves from Kurt Lohmüller's work that this does have an effect. And in simulations, when you have really strong, if you go from outbreeding populations to a really strong bottleneck, then you would expect to have reduction in fitness due to genetic load, but these are plants that were already inbreeding at a high level, so they probably purged a lot of the really deleterious variation that was around. Okay, so I'm going to tell you about a few of the traits. So we've done trait mapping in these populations, so flowering time and fitness, drought tolerance, and water use efficiency, and soil nutrients. These are volcanic islands, so it's something we were interested in. And so this is showing the growing season in Saint-Antal and Fogo at a couple of sites, and then the dry season, and you can see a big difference in the flora. So annual plants like Arabidopsis would be kind of trying to finish up their life cycles, potentially escape the drought. So we grew plants in these simulated CVI conditions and measured the time to bolting. So Moroccans mostly took a while and didn't finish bolting by the end of the experiment, where the plants were drying up and dying, whereas most of the Saint-Antal accessions flowered much earlier, and the Fogo accessions also. And we saw there was an inverse correlation between flowering time and fitness. So if you flower faster, then you have higher fitness in this environment. When we mapped the variation in Saint-Antal, we found a variant in Frigida that is a premature truncation of frigida, so it disrupts this frigida gene, which is in the flowering time pathway in this vernalization to cold part of the flowering time pathway. In Fogo, there wasn't a lot of variation in the population. There's this one, which is probably a contaminant, but we didn't know for sure, so we didn't remove it, but we don't see a lot of variation in the population. So we used a cross. We made a cross between a Fogo accession and a late flowering Thanantau accession. And then we did bulk segregant analysis to look for allele frequency dispersion, and we found one major peak, which is corresponding to FLC, and it's a truncation at the third amino acid in FLC. And so that's also in this vernalization to cold pathway. And this one's kind of interesting. Like I told you, Fogo seemed to have a small population size for a long time. And so we kind of think that this and some of the other variants that arose in that time are responsible for kind of rescuing Fogo from this constraint that it had. But it may not be the best way to adapt. Like you never really see FLC knockouts in Arabidopsis thaliana. You see variation that affects the like kind of non-coding variation in FLC that's affecting expression. That's really common, but you don't see complete knockouts. So we're not sure that there's a pleiotropic effect of it, but it's surprising to see this kind of variation. And so it could be that, yes, they adapted, they got faster flowering time, then they could flower early and escape the drought. Fogo is actually drier than Thanantau, and it's more recently active volcano, so it's a harsher environment in lots of ways. But maybe they adapted through a mechanism that wasn't the ideal mechanism, and that could actually kind of come back also later to be an issue, because you can't reverse from a knockout once you have a loss of function. So within the Cape Verde, we find in one island that we had frigida knocked out, or loss of function of frigida, and in the other one, a loss of function of FLC. So kind of a parallel loss of function events in the same pathway in the two different islands. And if we put this together with, if we reconstruct the history of the FLC region, it's kind of corresponding to the time when the Fogo population started to expand. So this and some of the other variants that arose in there, which I'll talk about, another one might might have allowed evolutionary rescue there. Okay, and then we also looked at water use efficiency. This was, so Cape Verde has this kind of foggy environment, and these humid trade winds come in from the northeast and feed fog to the high mountains, so it's got kind of a special sort of, even though it's got a short dry season or short growing season, during the growing season it also has this fog. So it means that they're not it's not an arid environment when in the growing season. Ahmed El-Faragi and my group was working on drought tolerance, and he collaborated with Olivier Lédé and Andreas Weber. Olivier has this set up to do phenomics of drought response. If it's a mild drought response, it's difficult to do an extreme drought response and get a good images that are reliable. So it's a mild drought response. And so this is showing the delta C13, so water use efficiency. And we find actually that Moroccans are the most, have the highest water use efficiency, whereas Thanantau has the lowest and Fogo is in between. And, but conversely, we see that the expansion rate, so the growth rate of the rosette is higher in, especially under well-watered conditions, drought conditions, under well-watered conditions in Thanantau and Fogo. So they seem to have this kind of fast growth, but they're not really, they try to escape the drought rather than to tolerate the drought. When we map it, we find one really strong peak in map K12 that explains 36% of the variance in water use efficiency. And yeah, and so I don't have anything else about map K12, but this is involved in stomatal aperture. So this variant in map K12 is has a similar effect to mutants of MAPK12, which cause stomata to be more open and less responsive. So under a really humid environment, then they would be able to photosynthesize more, right? If you open your stomata, then you can have a higher photosynthetic rates and faster growth rate. So together between flowering time and water use efficiency, both of these would push in the direction of this kind of fast life history escape strategy to grow quickly and flower before the drought really sets in. Okay, and then I'm going to give you a whirlwind tour of work that Manu Tergamina has been doing on soil nutrient levels, and this is in collaboration with David Salt. Okay, so Manu with David collected data conducted elemental analysis on 20 different elements, and this is showing a principal component analysis of that, of the leaf ionome, and you can see this is Fogo here and Sanantal in Morocco, and it's showing that you have many of these elements have a different accumulation rate in Fogo compared to Sanantal in Morocco. There's a little differentiation of Sanantal as well. So the CVI plants have a distinct leaf ionome. Both of these islands have a volcanic origin, so the soil could be different from what you find in the mainland. Fogo in particular has really recent volcanic activity. This is showing different times of eruptions. We have populations up here and a population down here. So our kind of lava population is down here, and then population in Inferno. So some of those are with the older eruptions, but they're still much more recent than the Sanantal eruptions, and would have been when the plants were the recorded ones, and the ones that you can see here were happening when the plants were kind of expanding across the islands. So this specifically, I'm going to talk to you about iron and manganese, and iron and manganese are both involved in photosynthesis, but in different parts of the photosynthetic pathway in photosystem 1 versus photosystem 2. And if you have an imbalance of iron and manganese, then you end up with a buildup of reactive oxygen species, and that can damage tissues. So it's important to not only have enough manganese and iron, but maybe even more importantly, to have kind of a ratio of iron and manganese that is like going to work for the plant and not cause tissue damage. Within Fogo, we saw different degrees of chlorosis. So some of the plants were sort of green, like kind of vibrant yellow-green, and others were very yellow and small. And this could be quantified, the greenness, and also the size here. And you see the Fogo, Sanantal shifted relative to Morocco, and then Fogo is shifted relative to Sanantal in Morocco. To try to understand what was the basis of this divergence in size, we used a population, an F2 population created between col 0 and this Fogo individual F13.8. And within the F2 population, we saw a really wide variation in leaf color and size. This is a distribution of the phenotype and the parentals. And then we sequenced just this tail, which was not a huge number of individuals, so we didn't have a whole lot of resolution. But like I told you, the variation in Fogo, if we're looking for variation that's specific to Fogo, there's not that much variation within Fogo. So we're looking for what causes this variation there. And so we did a bulk cyclogen analysis again, and here we find, so this is a region that was fixed for the col 0 allele frequency that was causing chlorosis, and a region that was fixed for the Fogo frequency that was, with the Fogo frequency that was causing chlorosis. And these map to two candidate genes that are both known to be iron and manganese transporters, but with different affinities for the two elements. So here we have this N-ramp and IRT1. And IRT1 is again, so maybe in the next, but these are both iron manganese transporters. Manu made a couple of CRISPR lines, starting with a Sana and Tau individual, introducing an IRT1 frame shift, so a loss of function of IRT1, and he kind of recapitulates this difference in phenotype, and if he maps it onto the PCA, then now this S1 Sana and Tau plant clusters together with the FOGO individuals, so that's kind of sufficient to stick it into that FOGO cloud. The IRT1 mutants had lower iron accumulation, and so that would have kind of rebalanced the manganese, and then they also have higher manganese accumulation. So here, I think I didn't mention it. So, so the soil in FOGO, the issue with the soil in FOGO is that the iron is, because the soil is kind of newer soil that's been produced from volcanic activity, then the iron's not as oxidized, but the manganese levels, the absolute manganese levels are very low. So the balance would have been high iron to low manganese. And so this is allowing them to transport less iron and more manganese just from this one change in IRT1. And so we expect this provides an advantage in this high iron and low manganese soil. So if we look at the distribution of IRT1 in the FOGO population, it's actually fixed. So similar to FLC, it fixed early in the population. It's not found on Sana and Tau or anywhere else. In fact, IRT1, normally an IRT1 mutant, it doesn't survive. So an IRT1 mutant in the call0 background doesn't survive unless you supplement with iron. So this is a really strong effect variant, and would not really be viable normally. So it's also another case where this might have helped to rebalance the iron and manganese in the plant, but it's not what you would choose to do if you, if you were going to decide how to adapt, if you were making the decision. We see a evidence for selection from a composite likelihood ratio test. So it's like a hard selective sweep where this region was driven quickly to high frequency, and that's still visible in the population, which, because of the kind of bottleneck that was happening, it was maybe, it's surprising that it would stick around the selection signal, because if the population size is low, then the amount of time that you expect a selection signal to stick around is not that high. We also, if we reconstruct, this is a coalescent tree for the region. And this is a reconstruction of the time when the selection coefficient, the change in allele frequency and a selection coefficient derived from that. So we get a 13% selection coefficient, and that's with a background population history of rapid growth. So so it looks like strong selection, and you can just kind of see that the populations are kind of not. You've got a little bit of Montevella here, lava's kind of split up in Inferno, and I'll come back to that in a second. Okay, so for NRAMP, we find that we have these this tandem duplication at NRAMP. So this other region turns out to be a tandem duplication. And when we look at the expression associated with the these call0 versus F13.8, we see higher NRAMP expression consistent with that being a tandem duplication that's causing upregulation. And so NRAMP 1 can provide an alternative route for iron transport. So this would have an effect back on the to increase the iron after maybe it was an overshoot from the first. So what's the distribution of NRAMP? Well, when Manu looked more closely, he found that there were 3 different tandem duplications and no evidence for recombination. So they all have different breakpoints. And so there's 3, it looks like 3 different tandem duplications arose in this region of the genome. And remember, this is a really young population. Maybe it's 4,000 years, the last 3,000 to 4,000 years. And so I think that's really interesting from the perspective of if you're looking at a much more diverse population, you might expect this kind of stuff could be happening often. So it looks like a kind of soft sweep of tandem duplications. These are 98% frequency across the island. So that would be like a soft sweep with the different haplotypes that arose separately. And then if you look at the distribution, they're somewhat structured in the distribution across the island. So you have different haplotypes. One haplotype is only found in one individual. And then we have a couple of single copy ancestral variants that we think might have something else that upregulates NRAMP1 a little bit. That's more cryptic, but it's not. It's got a similar level to the ancestral. Okay, and then when you look at when you reconstruct the coalescent tree for the region, then it splits more with the population structure. So Montevellas are kind of all together, and so it looks like it arose. What we think it happened is that the IRT1 fixed, plants started to spread. Maybe also the iron is becoming oxidized, so actually they start to need more iron, maybe, and then they start to get these in-ramp tandem duplications. Yeah, the copy number is associated with greenness and with leaf iron concentration. So when you have higher in-ramp copy numbers, then you get higher iron concentration. And it explains 87% of the heritable variance in leaf iron on the island. And then when we look at NRAMP1 expression, then we see an increase as we go to higher copy number. And when we reconstruct it, it's like we have a IRT1 knockout, and then we have these different NRAMP variants that arose kind of as the population was spreading across the island. Okay, so it's these plants experienced a sudden multivariate change in the environment. You have this kind of Fisher or geometrical model where you have it's similar to Cape Verde, where you have a population that's kind of wouldn't be a population with a lot of standing variation, right? A population that's bottlenecked best fits that model. And then you have this series of genetic steps. So you have the new colonist that came in, and it's not well adapted. It needs to, it's not close to the fitness optimum. And over time, it moves towards the fitness optimum through steps. And what we see with this, it's kind of a two-step model where we go overshoot with the IRT1 loss and then move back up to the fitness optimum with the tandem duplications at NRAMP. So I think I'm out of time, but from this last part, Manu has another paper that just came out this year where he's looking at boron, which is also something that's limiting in volcanic soils because it tends to stick to boron, tends to stick to it and be unavailable. It's absorbed by volcanic soils. He finds this BOR2, which is a SNP, and then he uses this double haploid population that we produced, finds again a structural variant, this time in BOR1, and again when he looks across the population, he sees three different breakpoints for BOR1. If he puts that in as a covariate, then he gets a stronger signal for the BOR2 SNP, so it helps to have that. And when he looks at the BOR2 effect, all these red ones have some kind of a tandem duplication. They have extra BOR1 copy. And so, again, it looks like something that spread as the population spread. There's a kind of higher frequency tandem duplications, but tandem duplications seem to be able to happen more easily than you would expect. You know, maybe part of it is where on the chromosome these guys are kind of subtelomeric on the chromosome where there might be a higher rate of tandem duplication, but it seems surprising for this small population that we would have such a pattern. Okay, and so both SNPs and structural variants are involved in adaptation, and the tandem duplications seem to show up a lot, at least in Cape Verde, where the soil environment is really different You get tandem duplications a lot, and that's also been found in other species. Sometimes where you have metal hyperaccumulators, it looks like tandem duplications are important there. So, and these kind of patterns might also be found in older populations, but be more difficult to see. So we start to gather up different functional variants, and then if we have CRISPRs, then we can also look at the pleiotropy directly from the CRISPRs and also the kind of concordance of peaks and GWAS. And we have different kind of populations with different histories that we can study. I've just told you about Cape Verde, but we have a lot of work going on in East Africa too. So within thaliana, there are these kind of understudied systems from isolated island populations to Afro-alpine populations. They're distinct from the mainland and the CVI populations are distinct from the mainland and can serve as a model for understanding evolution in island endemics. And the populations there have adapted to a new multivariate environment using new variants that arose after colonization. And so we can potentially understand from studying these how traits and molecular changes are linked to the environment. These are all mostly people who are gone, but were involved in some way in the work I just showed you. And I think I have this. We're not so much recruiting, but we're always interested in talking to people who might be interested in the lab. So, okay, I'll just leave there.